Hi there, my name is Salah Elbashir, and I am very happy to welcome you on board with Salah Elbashir Academy C. You join thousands of biology students who are already skyrocketing their knowledge with biology in our academy. There is just two tiny steps you need to take to achieve your amazing and marvelous goals. Understanding biology science and studying biology science hard. Now let us go to start our first step. Body tissues can be classified into four basic types according to their structure and function. And from here forward, I would like you to remember this statement. Always structure matches the function. The first category of tissues is called epithelium. The second is the connective tissue. The third is the muscle tissue. And the fourth is the nervous tissue. So those are the four categories of tissues. In this lecture, we are going to study the epithelial tissue or the epithelium tissue. The word epithelium comes from the Greek two portion, a prefix called epi that means upon or top, and thelium which has different meaning, but here we are talking about a cell layer. So it has different meaning like papilla or nipple, but the main that we need to know here is the meaning of cell layer. So thelium here means a cell layer. Epithelium is the singular word, while epithelia is the plural. Okay, so this is the main things. We are going within the epithelial tissue to know how this epithelial tissue is formed and how what is the epithelial tissue. We are going to do this within the class. Epithelial tissue consists of cells arranged in continuous sheets in either single or multiple layers. So it is a type of membrane cellular tissue that covers a free surface or lines, a tube or cavity of an animal body or human body as well, and serve especially to enclose and protect the other parts of the body to produce secretions and excretions and to function in assimilation. Their cells are closely packed with little intracellular space between adjacent plasma membranes and are held tightly together by many cell junctions. Epithelial tissue is an excellent protective barrier for body surfaces. And the main example of this is the skin. This tissue has specific characteristics. Cellularity, individual cells are closely attached to each other at their lateral margins to form tight sheets. And we say that in the definition first. It has little or no extracellular matrix. It is avascular, that means there is no blood vessels in this tissue. This tissue has no blood vessels, but it is innervated. That means this tissue can sense. It has polarity and attachment. So it's arranged so that there is an apical surface layer or free space or apical space, and also a basal region that is attached to the underlying connective tissue. Epithelial tissue also has tissue functions. So the tissue functions is the first one is the protection, second one is the absorption, secretion, third excretion, surface transport and permeability, and sensory function. So we have 6 functions. So in protections, it covers body and lines all body cavities, provides a protective shield, physical protective shield for underlying tissue. Example of this is the epidermis. Absorption in some parts of the body, it takes in certain materials. The best example for that is the digestive tract. Secretion in some parts of the body of the epithelial tissue is specialized to secrete certain products. Example of that in the small intestine. There is the epithelial of tissue of the small intestine, and some cells in it are specialized to secrete mucus, for example. And this is a secretion part of it. Excretion in the kidney, for example, cells rid the body of materials. As a transporter, there is a specific structures called cilia on the top surface. These cells in the lungs remove mucus and particles up and out. Be careful, in one direction to the outside. So it has no two-direction waving. It waves only in one direction from inside to outside. I will ask you a question. Why? Especially in the respiratory system. Why? It controls permeability. That means it allows some of the materials to get into the cell or outside of the cell and inhibits others. Sensory function. It has a sensory function like the taste buds in the mouth. So it has senses, and it could easily, you can taste within the taste buds, which are part of the epithelial tissue. Epithelial cells have a specific structure. And again, remember that structure always matches the function. So because of its structure, it performs a specific function. Let us take the cells here as a sample. This is a symbolic cell. It is not all types of the epithelial cells. They are not like this, all of them. But this is a symbolic one, just to know how the cell is looking and how its shapes look like. So any of the epithelial cell has an apical surface. So this is the apical surface of the epithelial cell that faces the upper surface. So here in this place we are going to see the free surface, where this surface, the free surface, always faces either the cavity, a lumen, or an outside surface like the skin. The upper part of these cells is facing uh, the outside environment. So this, it might be in the intestine, it might be in any hollow organ. So it faces either the lumen or the cavity, the free space. The second space, the second surface here is called the basement surface. This one, the basement surface, and because here this is the 3-dimension, we call it the basolateral. So because here there is another part of the or the remaining of the cell, and here is the front part. So this is the basement membrane, and this is the basolateral surface, which are very important to each other. The cell lies on the basement membrane, and actually it's germinated from this basement membrane. Then the apical surface may contain some structure, and those structure modify the function of the cell. Like what? Like this hair-like cilia. They are looking like hair-like structures, and they move in one direction. They don't move in two directions. And also there is another one look like some sort of uh, cilia called stereocilia. And also they might be microvilli or villi, and those are for different things. These cilia and stereocilia are for moving materials, while the microvilli is a plural name, the cilia is a plural name. So the microvilli may absorb things from outside to inside the cell. So this is the function. So when they are present in the apical surface or the top surface of the cell, that means this cell had ability to absorb materials from outside. Then we have the basal surface, as we said, which we call it the basement membrane. And here are the organelles of the normal cell, like Golgi body or Golgi apparatus, Golgi or Golgi apparatus. Here is the nucleus, and those are two types of mitochondria, and maybe there is other things like this one, vesicles, and these vesicles may contain different types of materials, or like the enzymes or something similar. So this is the main thing of the epithelial cell. Here is the side or lateral side of the each of these two cells, And those lateral sides are connected to each other with cell junctions. We are going, after a while, we are going to talk about these junctions and what are they, what their function is. But it's going to be just, when we move to them, we are going to see. Here, I just want you to be sure that those modifications of the surface, it might be without any of them. It might be with cilia only or with microvilli only. This depends on the what function is this cell should do. And this is the most important thing. So primarily, this is the cell, the epithelial cell, and how it looks like. Again, this is just a symbolic cell. Not all cells look like this. We are going to talk about the shapes of different epithelial cells just a moment. Regarding the characteristics of the epithelial cells, we talk about shape and size. So the shapes and size of the epithelial cells are variable, ranging from tall columnar to cuboidal to low squamous. And squamous means flattened. It looks like the scales of the fish. It is scale-like appearance. The cell size and morphology are generally based on their function. So here again, I would like you to understand and apply in all your biology science the statement that says structure matches the function. Polarity is the second characteristic. We know that the epithelial cells are generally show polarity with organelles and membrane proteins distributed unevenly within the cell. The apical, or the free surface of an epithelial cell, is present towards the body surface, the body cavity, the lumen of intestinal organ or a gland, duct that receives cell secretions. Apical surfaces may contain cilia, stereocilia, or microvilli, and the three of them are modified structure to perform a specific function. The lateral surfaces of an epithelial cell facing the adjacent cells on either side may contain intracellular adhesions and other junctions, and we are going to talk about this. The basal surface of an epithelial cell adheres to the extracellular materials such as the basement membrane, which is an inert connective tissue made by the epithelial cells themselves. Be careful. It is made by it is a connective tissue, but it is made by the epithelial cells themselves. Let us see the next slide. Continuing with the characteristics of the epithelial cells, basement membrane, which is one of the most important characteristics of the epithelial cell. The basement membrane is a thin extracellular layer that commonly consists of two layers. Be careful here. The basal lamina and the reticular lamina. Be careful. The basal lamina is closer to and secreted by the epithelial cells and contains proteins like laminin and collagen, as well as some glycoproteins and proteoglycans. The second layer, again, be careful, the reticular lamina is closer to the connective tissue, present underneath, and contains collagen proteins produced by connective tissue, not the epithelial tissue, connective tissue cells called fibroblasts. Fibroblasts are cells, and those cells belongs to the connective tissue. They are not belongs to the epithelial tissue. Be careful. One of the most important characteristics of the epithelial cells are the renew and repair. Epithelial cells have a high rate of cell division, which allows the epithelial tissue to continually renew and repair itself by sloughing off dead or injured cells and replacing them with new ones. And it is it's really very important characteristics, because some other tissues may cannot perform renewing and repairing. The intracellular adhesion and junctions are contact points between the plasma membranes of the adjacent epithelial cells that are tightly joined those cells into a functional units. And here in this figure, they are arranged orderly from up downwards, so we can see what type of junction is there. So here on the top in the free surface, this is the free surface or the apical surface of the cells. Here is cell B and here is cell A, and those are the adhesions and junctions. Here is the tight junction at the top. Then the adherence junction here with the actin filaments. And then after that is the desmosomes, or are the desmosomes, and they have an intermediate filaments, connexin. And here is the gap junctions with the connexins. And then here is the this one and this one, the hemidesmosomes here and focal adhesion. They are different. You see the color is different, so they are different types. So those are how they arrange within the cells and how they tie those two things together. They are important for adherence, and at the same time they play other roles like the gap junctions, which mediate transport of fluid between the two cytoplasms. And let us go for the second slide. We can see, or the next slide, we can have more information about them. Continuing the intracellular adhesion and junctions, we have two columns here. One for the intracellular connections, the other is for intracellular junctions. Those are for support and communication. The intracellular connection, the most important function for them. Number one is called cell adhesion molecules, and those this is the acronym of them, CAMS, CAMS, transmembrane protein, which is a transmembrane protein, so it is present in the membrane. Here is the intracellular Siemens, so this is between the cells, and it is a proteoglycans. Chemical nature of it is a proteoglycans. It's made of protein and carbohydrates. And here is hyaluronan, and this is glycosamine no glycans, and they are always comes after the tight junctions in order. When we take them in order from up down, they are coming always after the tight junction. Now we have three junctions here. The tight junction, which is at the apical junction at the top, where we can find them in the apical surface. Between two plasma membranes, adhesion pelt attached to the terminal web, prevent passage of water and solutes, isolate waste in the lumen. Desmosomes comes after this. CAMS, it made of this one, dense area, and intracellular Siemens. So it has a intracellular Siemens. It's formed of this one. And we have two types, the spot desmosomes and the hemidesmosomes. Those tie cells together and allow bending and twisting of the cell. And this is the hemidesmosomes. They attach cell to the basal lamina. Be careful when we go. When you go to the upper image, you can see them and see where they are and what they function for each place. The gap junctions allow rapid communication, so because they allow passage of ions, here they allow ions to pass, are held together by channel proteins, junctional protein, connexon, so they have a connexons. And this is a channel protein. The most important thing of the gap junction is that it allows the passage of ions. Ions are electrically charged, so be careful about that. It also allow the charge to move from place to place, coordinate contraction in the heart muscle. And this is why we say that it has an electrical appearance or electrical energy, because it allows the passage of ions, which is very important here. This is all about intracellular adhesion and junctions. Epithelial tissue, or tissues actually, is classified based on its anatomical characteristics and physiological characteristics. So we have two main classification. The first one is based on its anatomical or structural. We say anatomical, that means structural. And the second is based on its physiological or functional characteristics. And this gives us two types of classification. We are going to talk more about them in the next slide. Epithelial tissues can easily be classified according to their anatomical characteristics or physiological characteristics. That is to say, their structural characteristics or their functional characteristics. So we can easily have two big subcategories of classifications. Based on the anatomical, structural classification, covering and lining epithelial tissues are classified according to two characteristics: the shape of the cells of the apical layer and the number of layers the cells are arranged in. So here we have three main shapes of cells that are could be seen in the apical layer. The first one is called squamous, which is a thin, centrally nucleated cells that allows substances to pass rapidly through their membranes. And that is why they are flattened, and that is why they have central nucleus, and that is why they are thin. So they are present in a place where exchange of materials is quickly and rapidly and necessarily. Remember what I have said, the statement that says a structure matches the function. The second type of cells are called cuboidal cells, which are square-like. They are a little bit wider and it has height. It looks like a square or a cube, and they may or may not have a microvilli on the top of its apical layer. And if they have this microvilli, that means they are important, or they function as an absorptive cell. So they absorb materials from outside to the inside. If there is no microvilli on the top or the apical surface, that means they are important for secretion. So these cuboidal cells are important for either secretion or absorption. The third one is the columnar one, or columnar cells, and those are much taller compared to the previous two, and they are rectangle shape. That means their height is more longer than their widths. And I would like to mention here something, that they have also a prominent nuclei, but their nuclei are launched on the bottom third, okay, of those cells. They may have cilia up in the apical surface or the free surface of the cell, or microvilli. So if they have a microvilli, again, that means they are cells that are responsible for absorption. But if they do not have it and they have only cilia, that means they just move materials from outside. And again, cilia are one directional waved structure. Also, it might not be none of these, so may have a secretory function. So it is either for transporting material or absorption, or also has a secretory function or secretions. Okay, I bring this here to be clear for you. This one is called pseudo-stratified. Pseudo, this is a word that is formed of two portions. The prefix pseudo, which is a Latin word that means fake, and suffix stratified, which means consists of more than one layer. Again, these cells are in fact, columnar cells. The only problem is that there are two types of columnar cells. One of them is dominating the other because it is taller than the other. So it appears as if there are two different types of cells, which is not. Both of them are columnar cells. Again, there is another characteristic which is very important, that the nuclei are arranged in two rows. Why? Because of the difference of the height of each of the two cells component, the short cell and the top cells. So it appears as if there are two rows. And because of this appearance of those two rows, it appears as if it is formed of two layers or consist of two layers, that the cells are arranged in two layers, and gives a fake appearance of a stratified tissue, which is not. It is not an stratified tissue. It is not a stratified tissue. It is just a single layer tissue. And because it is a single layer tissue and made of the same cell, it is called pseudo-stratified, fake multiple layer tissue. It is fake. It is not that. Okay, how could I know that under microscope? Just look to the basement membrane, and then you are going to see that all these cells emerge from the same basement membrane. This will tell you that this is fake. A fifth type of transitional cells called the transitional cell. We are going to talk about it very clearly in the next slide. And it's called transitional. So it has two appearance. It transitional. It transfer or change from shape to shape. Why? Because the importance of this cell, or the function of this cell, is to allow relaxation and distension. It could easily tolerate the distension by changing its shape, and the distension is due to that the organ had the ability to distend. Like where? Like the urinary bladder. You see, when it's filled with the bladder, or the bladder is filled with the urine, it's going to distend. So in order to have a room, these cells change their shape, and this is called transitional. We are going to see that more clearly in the next slide. So this is the first characteristic, that the shape of the cells of the apical layer. The second characteristics, which is very important, is the number of layers and how the cells are arranged in those layers. So the arrangement of the cells in layers, how many layers are there? If it is a single layer, it's called simple epithelium. If it is a multiple layers, we call it stratified epithelium. And we are going to see more better, the vision is going to be more clear in the next slide. Now we are going to talk about the anatomical or structural classification. And also we are going to answer how epithelial tissues are named and classified after their anatomical classification. As we said, that there are two more important points we have to consider when we are talking about the naming of the epithelial tissue. First is the shape of the cells of the apical layer. Second is the arrangement of the cells in layers. So based on those two facts, we are going to name them. The big categories is when we have one single layer, we are going to name it simple. When we have more than one layer, like this one, more than one layer, we are going to name it stratified. More than one layer means more than one. So 2 is stratified, 3 layers are stratified, and so on. Now, let us go to connect the shape of the apical cells with the number of the arrangement in cells, arrangement in layers. So here on the top layer we have a squamous epithelium because it is flat, centrally nucleated cell. This is the two lamina. This is the basement membrane, and here is the connective tissue. So here we have the blue is the basement membrane, and you know that the basement membrane has two laminae, the basal lamina and the reticular lamina. This is not our consideration here. Here, this is the full blue line is the basement membrane. Above the basement membrane, there is only one layer, so it is simple. And the shape of the layer is squamous. So this tissue is named simple squamous epithelium. Simple squamous epithelium. Let us go for the second one. This one. What are the names here? What is the shape of the upper layer cells or the apical layer cells? So the apical layer cells are square-like. That means scuppidal. This is the line that shows the basement membrane. It is one line. All of them rise or emerge from the same basement membrane, and they are arranged in one layer. So this is one layer. It's called simple, and the shape is square-like or cupoidal-like. So it is cupoidal. So this is simple cupoidal epithelium. Let us go for number 3. The cells are arranged in one layer because this is the basement membrane. All of them arranged from emerge from the same basement membrane. So it is only one layer here. This cell is a rectangle in shape, so it is columnar. So because it is one layer, it is simple. And because the shape of the apical layer is columnar, so it is simple columnar epithelium. Now, I'm going to avoid talking about this just a minute, for just some moments, couple of minutes, and going to skip number 4 to number 5. So here, this is the basement membrane. How many? There are more than one layer. 1, 2, 3, 4, 5 layers, and 6 layers. So those are more than one layer. So it is stratified. It is not as simple because it is not one layer. It is more than one layer. The upper one, the apical one are formed of squamous epithelium. So the name of this tissue is stratified squamous epithelium. Go for the number 5, number 6 here. It is more than a layer, so those are 4 layers. All of them above the basement membrane, and each of them is different from the other. They are arranged in rows, so they are more than one layer. So since it is more than one layer, it is stratified, and because the upper layer is made of square-like cells or cube-like cells. It is cuboidal, so it is a stratified cuboidal epithelium. Number 7 actually, it is more than one layer, 1, 2, 3 layers. And since it is more than one layer, it is stratified. And because the upper layer here is made of a rectangle cells, the cells that their height is more wide or longer or wider than the width. So this is a rectangle or columnar cells. So the name of this one is called stratified columnar epithelium. Now we have number 4 here and number 8 here. Those are important. If you just go back and refresh your memories, we talk about this type of columnar cells that are two types of cells. One of them is taller than the other, and they have nuclei that are arranged into rows, which gives you the fake appearance of a stratified tissue. It is not stratified. How do we know that? We just go to the basement membrane. All these cells are emerging from the same basement membrane. They don't have two basement membranes or different basement membranes. So this is a single layer. It is not a multiple layer, and the appearance is fake. It is not a stratified, it is a simple. So this is a simple one. It is not a stratified, and this is why we put it here with the simple group. And because it's fake, we call it pseudo, and it is fake multiple layer, so we could call it pseudo stratified epithelium. But it belongs to this simple one. Now here, if you remember, we said that we have a cell that's called the transitional epithelial cell. And this transitional epithelial cells, we name it transitional because it changes its shape depending on the physiological status of the organ. What we do mean by the physiological status. Let us take in consideration the gallbladder. So, the urinary bladder. So if the urinary bladder is empty, that means the organ has nothing inside it to stretch. It has nothing to do with stretching. It is in the relaxed phase. So the cell take its normal shape, which is a cuboidal one with rounded apical surface. But when urine accumulates, it's going to press on this one, and since it's going to press on it and the organ itself distend, this will change. This cell is going to change shape and appears as if it is squamous, flattened. Why? Because it needs to distend to give room for more blood to get into it. Sorry, for more urine to get into the urinary bladder. So this is why we call it transitional epithelium. Okay, why we put it with the stratified one? Because actually here there is more than one layer of cells. So because it's more than, the cells are arranged in more than a layer, we call it stratified. And because the apical layer cells change their shape depending on the physiological status of the organ, we call into relaxed and stretched shaped, and this is why we call it transitional. Now it is very clear to you how we name the covering epithelium, especially the covering epithelium, depending on two facts or two statements, how the shape of the apical layer look like, and how those cells are arranged in layers. Nobody is going to be asked about the anatomical structural classification of the epithelial tissue without knowing those two facts. The shape of the cells of the apical layer and the arrangement of cells in layers. Those are the two facts that we depend on them. Be careful. Here we are going to study each of the two groups of the covering and lining epithelium, the simple epithelial tissue and the stratified epithelial tissue. And we are going through each category or each tissue and see where we can find it, and also the function of the tissue. So the simple squamous epithelium, we can find them or find it in the air sacs of the lungs and the lining of the heart, blood vessels and lymphatic vessels, glomeruli of the kidneys, and the serous membranes. The function of the simple stratified epithelium is the diffusion and absorption. Diffusion allows materials to pass through by diffusion and filtration and secrete lubricating substances. Absorption is also part of the function of this one. So you know the name and how it comes from where, what is the base of this name, and also you know where you can find it in the body and what is the function or of this epithelial tissue. The second one is the simple cubital epithelium. We can find them in the ducts and the secretory portions of the small glands and in the kidney, tubules, in the liver, pancreas, thyroid follicle, seminiferous tubules, and the germinal epithelium of the ovaries. Okay, what is the function of it? The secretion, absorption, and excretion. Remember, whenever we say absorption, most probably we are going to have some sort of microvilli. Simple columnar epithelium lines the stomach, intestine, gallbladder, and also the upper respiratory tract. Secretion, absorption, and excretion are the function of this one. So I want you to put in your mind how the structure matches the function in each of these so you can easily understand what is going on. Now the pseudo-stratified columnar, I added the word columnar here in order you remember always that this pseudo-stratified is actually a modified form of simple columnar epithelium to act partially in specific places. And this also will take you to this one. The structure matches the function. Ciliated tissue lines the trachea and much of the upper respiratory tract, as well as fallopian tube of the uterus. Here we add the word ciliated. It comes up. So what do we mean by ciliated? Ciliated means it has cilia on the top on the apical part of it. So since it had a cilia, that means it is responsible for movement of something. So in the upper respiratory tract and the trachea, it is responsible for expelling the mucus outside of the trachea. The mucus and the dirt, you see, and this is why you cough when you have something and when you have mucus. So it takes it out from inside the body to the outside of the body. For the fallopian tube, it also it grabs the egg from the ovary to inside the uterus, and it has another one-way direction from the ovary to inside the uterus. Stereocilia, which is some sort of cilia tissue in the epididymis. Epididymis is a part of the male genitalia, and also it is important there to move the sperms and push them towards outside. And non-ciliated in the ductus deferens. Also, there is a non-ciliated pseudo-stratified columnar epithelium in the ductus deferens, which is a part of the male genitalia, and they are important there. And since it is non-ciliated, that means it had nothing to do with movement of something, of materials. Okay, what is the here we can say secretion, absorption, and excretion. In addition, or as well as transportation or movement of materials. So this is the part of the simple epithelial cells. Moving through the stratified epithelial tissue, the stratified squamous epithelium, we can find that in as a non-keratinized or keratinized stratified squamous epithelium. What do we mean by keratinized or non-keratinized? Keratin is some sort of protein. It is a dead part, and it's secreted by the upper cells, and it remains there. It provides more protection, and also it is waterproof. So the non-keratinized lines the esophagus, oral cavity, pharynx, larynx, vagina, outer layer of the cornea. Keratinized, we can see it clearly in the skin. You see all our skin. Both of them, the non-keratinized and keratinized, function as a physical protective barrier. So they have the function of physical protection. Stratified cubital epithelium, we can find them in the sweat glands, salivary glands, mammary glands, and surrounds an ovarian follicle. It has also physical protection, and also it has a secretion function. Stratified columnar epithelium, you can find them in the male urethra and the ducts of some glands, the conjunctiva of the eye, large ducts of the excretory glands, and it has a secretory function plus a physical protection. Transitional epithelium could be found in lining the urinary bladder, urethra, and the ureter, as well as the renal calyces. It allows the urinary organs to expand and stretch. And you remember what we talk, our talk about the transitional epithelium. Here we are going to talk about the modified epithelium. First, is it a new type of epithelial classification? No. Is it a new epithelium that differ from the others? No. So why it is there? Because there is a little modification in this epithelium to perform a specific function. And this is why it has maybe a different name, and also it is specifically found in some places. And here we are going to talk about it one by one. So the first modified epithelium is called mesothelium. It comes or originates from the simple squamous epithelium. So it has all the characteristics of simple squamous epithelium. It lines the body cavity like the pericardium, the pleura, the peritoneum, and so on. It is for physical protection. This is the function of protection, and controls the permeability. So it's named the mesothelium because it's found in those places and it performs a specific function. The second one is the endothelium. It is also a simple squamous epithelium. It originates from the simple squamous epithelium. It had all the characteristics of squamous epithelium. The only difference that it is the simple squamous epithelium that lines blood vessels and heart. So it is specifically found in lining the blood vessels and heart. Okay, can we see epithelium in the mouse that covering the mouse or even covering our skin? No. It is specifically named endothelium because it is found in the circulatory system, specifically in blood vessels and heart. Also, it is for physical protection and controls permeability. The third one is alveolar epithelium. It originates from simple squamous epithelium. It had the same characteristics of the simple squamous epithelium. All these characteristics of simple squamous epithelium. The only difference that they lines the alveoli of the lungs. So because of its location and because in this location it has a specific function, then it is separated to be named alveolar epithelium. It is a modified epithelium. How it has it function as physical protection, second gas exchange, third controls permeability. The fourth one is called cupidal epithelium with microvilli. We say that microvilli is a modified structure. It is the same structure, it is the same similarly like simple cupidal epithelium, originates from the same one, but it lines some parts of the digestive tract, and because of this it has additional function of absorption and physical production. So for this reason it is a modified with microvilli. Okay, let us go forward. Moving forward within the modified epithelium, we have the simple ciliated, columnar, epithelium with or without goblet cells. So this originates from simple columnar epithelium. It lines the nose, trachea, bronchi, fallopian tube, and uterus. It has a function of physical protection, move fluids from surface to surface, secretion, mucus, and enzymes. And this is why the goblet cells are there, because goblet cells actually are glands, unicellular glands that are important for secreting some mucus or sometimes enzyme depending on where they are. Then here we have a pseudo-stratified ciliated columnar epithelium with goblet cells. This it comes from, or the main original epithelial tissue is the pseudo-stratified columnar epithelium. It is found in the upper respiratory tract and also for the same things, that physical protection, move fluids from the surface, and secretion of mucus and enzymes. Sensory epithelium. Sensory epithelium, it has something new here. It's a stratified columnar epithelium. It is found in the retina of the eye, microvilli, taste buds on the tongue, that microvilli, organ of the corti, stereocilia, and ampullae in the inner ear, microvilli, stereocilia, and cilia. And this is for physical protection, gas exchange, and controls permeability, and also for taste, sensation. Let us move forward. The second type of classification, the big classification, is the physiological functional classification. And here it also split or divide the epithelial tissue into two big categories, the glandular, and this is what we are going to talk about, and non-glandular covering epithelium, which already we have talked about it or explain it as when we talk about the structural or anatomical classification. And here I wouldn't like you to make a problem. The non-glandular epithelium is what already we have been talking the previous structure. It's the simple squamous, the two types, the simple and the stratified, the simple squamous, the stratified squamous, the simple cubital, the stratified cubital, the simple columnar, the stratified columnar, and the pseudostratified and transitional. All these 8 are called non-glandular or covering lining epithelium. So what we are going to talk about is the glandular one, this one. But this classification is based on the function, because this is the main function of the non-glandular epithelium is to protect. It has also the functions, the other functions, but the main one is the protection. This one is the glandular. It has different ones. We will see what are they. Glandular epithelium. Epithelial cells that function mainly to produce and secrete various macromolecules may occur in epithelia with other significant functions or comprise a specialized organ called glands. Scattered secretory cells, known also as unicellular glands, are common in simple cubital, simple columnar, and pseudostratified or epithelia. Glands develop from covering epithelia in the fetus by cell proliferation and growth into the underlying connective tissue. So they do not grow upwards, they grow downwards into the underlying connective tissue, followed by further differentiation. So what we meant by differentiation? Differentiation means that there is a stem cell that has no specific function, and these stem cells, by time, when at the fetal level, they are going to change themselves and perform a specific function. This is called differentiation. So differentiation is a development process from a stem cell into a specified functional cell. How we classify glandular epithelium? We have 4 types of classification, each one based on specific characteristics. So classification based on the presence or absence of ducts. The first classification, the first type of classification is based on whether there is an absence or presence of ducts in the gland. The second type of classification is known as classification based on modes of secretion. The third one is classification based on the type of secretion. And the fourth one is the classification based on the number of cells. So we are going to go through each of them and see how each of them classify the glandular tissue, classifies the glandular tissue. Now, the first classification is based on the presence or absence of ducts. Now, we have specific criteria or characteristics in the first column in your left, and then we have the two classification. So, based on the presence or absence of ducts, we have 2 types of classification, 2 types of glands, either endocrine glands or exocrine glands. So the first one is the presence or absence of ducts. So presence of ducts, in the endocrine gland, there is no ducts, or they call ductless glands. And here they are with a duct or with ducts, depending on the type of the exocrine gland. The type of secretion in each one, here it secretes hormones, here other than hormones, chemicals other than hormones, examples are enzymes, the saliva, the semen, etc. Destination, we mean by the destination where their chemical acts. So the destination is far from the glands via blood. So this gland secretes, it is the endocrine glands secretes their hormones into the blood, and they act on a target tissue away from the gland itself. It is not nearby or in the vicinity of the gland. Okay? While in the exocrine gland, it is different. The secretions, whether it is enzyme or not, it goes or transported via the duct into an organ that in the vicinity of the exocrine gland, like the intestine or oral cavity, and through the gland ducts. Example of each of these is the thyroid gland. It is an endocrine gland. The parathyroid glands, they are endocrine glands. The testis is an endocrine gland. The ovaries are endocrine glands. The pituitary gland, the adrenal glands, the pancreas. Be aware. I put the pancreas in red. Let us see what is going on. Here is like the salivary glands, the intestinal glands, the pancreas. So the pancreas comes in the both of them? Yes. Be aware. Pancreas is an endocrine and exocrine gland at the same time. Now, the second type of classification is based on the modes of secretion. So I have two criteria here, and there are three types: merocrine secretion, apocrine secretion, holocrine secretions. So those are the type of glands depending on the mood. So the site of production and or release. The site of production is the Golgi apparatus or Golgi body, and it's released by vesicles, exocytosis. Here, the apocrine, the site is Golgi apparatus or Golgi body, and released by shedding cytoplasm. In the holocrine, it is released by cell bursting, killing gland cell. Gland cells are replaced by stem cells. Example, this is in the sweat gland. Here is the mammary gland. Here is the sebaceous gland. The third type of classification is based on the types of secretions. So I have also another three types of glands: the serous glands, the mucous glands, and mixed exocrine glands. And I have two criteria: the type of secretion. Here, the watery secretion. This is the mucus secretion, and here is post-serous and mucus secretions. Example: the serous one are parotid and lacrimal glands. Here is the sublingual glands, and this is the submandibular glands. Put in mind that sublingual and the submandibular, both of them are salivary glands, but one of them release or secretes only mucus. The other one secrete both of them, mucus and serous fluid. The last but not least is the classification based on the numbers of cells. So I have only two types of glands: unicellular glands and multicellular glands. Here I have the number of cells. I have only single cell or one cell. Here more than one cell. Structure of the duct within the cell. Here either simple undivided or compound divided. We are going to see this in the next slide. Shape of the secretory cells, secretory portion of the cell, not the duct, the secretory portion. Here is about the duct. If you are talking just about the duct, it's either simple, and that means undivided duct, or compound undivided or divided ducts. This is because of the duct. But here we are talking about the secretory portion, the other portion of the gland, which is the secretory portion, the portion that secretes the chemical. And here it is either tubular. You can't find difference here because it is only one cell. But here you can find difference. Either it's tubular, this is tube shape, or alveolar or acinar, which is it has a pliant pocket. Now, the relationship between the two, the ducts and the glandular area, so the relationship between the two of them, this one and this one, here per se there is nothing because it is just one cell, but here it is branched, so there is more than one part. Several secretory areas share one duct. Example of each one. For this is the goblet cells. So you remember mucus or goblet cells. We are talking about the goblet cells before. We did talk about them. Mucus or goblet cells are the only unicellular exocrine glands lining intestine, nose, trachea, bronchi, and so on. Here we are going to see the other one, so you can see the examples of each one, because they are more than one, and we have to make a combination between the duct and the tubular one. So we are going to the next slides. Now here we take about the simple glands, and when we take about simple and compounds, we are talking about the ducts, simple and compounds. And when we talk about the glandular part, this is another thing. We are going to say tubular, branched, alveolar, and so on. So here we have 5 simple and 3 compounds. The first one is called simple tubular, and we found them in the intestinal gland. Simple coil tubular, we find them in the merocrine sweat glands. And simple branch tubular in the gastric glands in the stomach, and mucus glands in the tongue, esophagus, and duodenum. And then this simple alveolar or acinar, not found in adults. Be careful, this is not found in adults, and it is a stage of development of the simple branch glands. Simple branched alveolar, this is a sebaceous or oil glands, and those present in the vicinity or in the neighborhood of each hair follicle. The compound glands here, we have a compound tubular. So this is the mucus glands in the mouse, the bulbourethral glands in the female reproductive system, testis seminiferous tubule, all of them are compound tubular glands. There is the compound alveolar or acinar glands in mammary glands, and we have a compound tubulo alveolar. This is in the salivary glands, glands of the respiratory passages, and in the pancreas. Thank you, all of you, for listening and for treasure attention. I hope that I declare everything and explain all about epithelial tissue. And I hope that you enjoy this session. I'm excited to see you in the next one. Thank you. Bye-bye, and have a good one.